Nerve Conduction Study, Part 3. Sensory Nerve Conduction Studies. Definition. Sensory NCS evaluates sensory axons by stimulating a sensory or a mixed nerve, recording the transmitted potential from another site along the same nerve. The recorded response is called sensory nerve action potential. Sensory nerve action potential are true nerve action potentials. Snaps are considered true action potentials because they represent the direct summation of electrical activity traveling down the sensory axon themselves. In contrast, compound muscle action potential are not true nerve potentials. They are muscle potentials generated by electrical activity of muscle fibers activated by motor axons. Types of sensory studies. 1. Antidromic study. Here, impulse travels towards sensory receptors. There is higher sensory nerve action potential amplitude and it is most commonly used clinically. 2. Orthodromic study. Impulse here travels away from sensory receptors. It has lower amplitude responses. Here, averaging may be required. Averaging in SNAP measurement, part of the nerve conduction study. It is a digital signal processing technique used to boost the signal to noise ratio, ensuring that low amplitude sensory signals are accurately recorded and distinguishable from the background noise. Difference between antidromic and orthodromic. Direction. Antidromic towards receptor, snap amplitude is higher, ease of recording is easier. Orthodromic direction away from the receptor, snap amplitude is lower, ease of recording is more difficult. Recording orthodromic snap is difficult. Recording orthodromic sensory nerve action potential is generally more difficult than antidromic recording because the resulting signal are smaller in amplitude and more sensitive to technical factors such as tissue depth. Why snap amplitude is larger in antidromic studies? Because recording electrodes are closer to the nerve and lesser signal attenuation. Motor artifact in antidromic studies. Motor axons may also be stimulated because their threshold is similar to sensory fibers and this can produce muscle action potential that obscure the snaps. Fortunately, sensory fibers conduct 5 to 10% faster than the motor fibers, so snap can still be identified. Methods of obtaining snaps. Pure sensory nerve studies, example for sural nerve and radial sensory nerve. Mixed nerve stimulation with distal sensory recording, example antidromic median sensory study, antidromic ulnar sensory study. 3. Distal sensory stimulation with proximal mixed nerve recording. Example, orthodromic median sensory study, orthodromic ulnar sensory study. Parameters measured in sensory NCS. 1. Snap amplitude. Definition, measurement, baseline to negative peak or positive peak to negative peak. Unit, microvolts. Significance reflects number of functioning sensory axons. Sensory latency, defined as time from stimulus artifact to the onset of snap. It can be peak latency, measured to negative peak. It is easier and more reproducible. Onset latency, measured to beginning of the snap, reflects fastest fiber better. Important points. Peak latency is easier to define and more precise clinically. Onset latency is better for conduction velocity calculation. Sensory conduction velocity formula, single stimulation site. Sensory conduction velocity equals distance by onset latency. Formula, two stimulation site, distance by proximal latency minus distal latency. In this figure, we can see antidromic median sensory nerve conduction study after stimulation at the wrist, revealing peak and onset latencies and sensory nerve action potentials. Amplitude, duration, and area are mentioned. The shock artifact interferes with accurate measurement of onset latency, whereas peak latency is easily measured. Important Viva points. SNAP stands for sensory nerve action potential. Antidromic studies produce larger amplitudes. Sensory fibers conduct faster than motor fibers. Peak latency is easier to measure. Onset latency is preferred for conduction velocity. Sensory amplitudes are measured in microvolts.